This video is going to focus on the use of enzymes. It's going to look at the different uses of enzymes and some of the different experiments that can be done with them.
The first thing we're going to look at then is invertase. Now invertase is an enzyme that's used to make sweets. Now it does this by taking sucrose and converting it, breaking it down into glucose and fructose, both of which are much sweeter than sucrose itself. Now the sucrose comes from sugarcane, and the invertase is naturally produced by a yeast called Saccharomyces cerevisiae. The benefit of all this means that you need less sugar overall because you're breaking down one large one into two smaller ones, and it produces lower calorie foods. The second enzyme we're going to look at then is the enzyme that's used in biological washing powders. Now there are two types of stains, soluble ones and insoluble ones. The difference in the two being that soluble ones wash out nice and easily, but insoluble ones don't, and they contain insoluble chemicals like starch, proteins and fats which don't wash out with just water. Therefore, biological washing powders have enzymes in them that help to break down those stubborn stains. So in the example of carbohydrates, which are example jam and chocolate, they'll use the enzyme amylase. Now that amylase will break down the carbohydrates into simple sugars which will easily wash out. Lipids, so for example butter and oil, they will use lipase, and that will break it down into fatty acids and glycerol. And finally protein, which for example is blood or grass stains, they use protease, and that will break down the protein into amino acids, which are again much smaller and can be washed out. One of the major advantages of using these enzymes then is that biological washing powders can be more effective at lower temperatures, so around 30 degrees, which is better than non-biological washing powders, and they also work best at pH 7, which is neutral, which is usually tap water. The third use for enzymes then is chymosin. Now chymosin is something that's used to produce cheese. The enzyme itself then, so chymosin, is usually found in the stomachs of calves, and it's found in a substance called rennet, R-E-N-N-E-T. So what they'll do is they'll take that out and they'll put it into milk, which helps to actually clot it, which starts to turn it into cheese. One of the main issues with this then is the fact that vegetarians themselves may not want it taken from a stomach of a calf. So what they'll do instead is they'll make it, and they'll make it from microorganisms, and you should be familiar with that process by now. So genetically modification, and then the rest of the process is exactly the same. The second part of this video then is going to have a look at different enzyme experiments. Now the first thing you need to know is what an immobilized enzyme is. Now normally when enzymes are used, they end up dissolved in the mixture and can be quite difficult to remove. So what they'll do is they'll use something called an immobilized enzyme. They don't need to be separated out because usually they're stuck to some sort of inert material, they're trapped and they can't move. So a key example of where this is used then is making lactose-free milk for people that are lactose intolerant. And that's what I'm going to go through now. So milk contains a sugar called lactose. Now normally that's broken down in the digestive system by the enzyme lactase, and it turns it into glucose and galactose, which are then absorbed into the blood. However, for people that are lactose intolerant, they don't have the enzyme lactase in their body, which means if they drink the milk, the lactose doesn't get broken down. This means that the bacteria in the gut can feed on it, and it can cause abdominal pain, wind and diarrhea. So this is where immobilized enzymes come in. So to produce lactose-free milk, they use an immobilized enzyme called immobilized lactase, and this is how it's done in the lab. So the first thing they need to do then is actually immobilize the lactase. So they'll take a solution of sodium alginate and they'll put the lactase enzyme together with it in a syringe. They'll add the mixture 1 drop at a time to a beaker of calcium chloride. Once they've done that, beads will start to form, and the lactase itself gets immobilized, gets trapped in those beads. If you leave them to harden for a few minutes, and then you can use any type of strainer, tea strainer, which will separate them out from the calcium chloride solution. So the second step is set up a column of these immobilized lactase beads. So they'll put some nylon gauze at the bottom of a column, and they'll put the lactase beads into it. There'll be a tap at the bottom of as you can see in the diagram that I'm drawing here. Once this is prepared, you can then put the untreated milk through it, and it will treat it and turn it into lactose-free milk. You can test to make sure the milk has been made lactose-free by using something called a glucose test strip. Start off by getting some of the untreated milk, which should have lactose in, and the treated milk that shouldn't, and place a glucose test strip into both of them. Now, because the lactase should have broken down the lactose into glucose, you should find out that the treated milk will have changed colour, and then the untreated milk will stay the same. This final part of the video then is going to have a look at the enzyme that's used to extract apple juice, and that enzyme is called pectinase. So pectinase breaks down pectin, which is a substance that's found in the cell wall of fruits. Once it breaks down this pectin, it allows the cell wall to release the juices. So to do this, the first thing you need to do is take an apple and chop it up into small chunks, and then put it into 2 separate beakers. Secondly, take some of the pectinase solution and add it into one of the beakers, and then put some water into the other beaker so it can be used as a control, and then stir them both. Incubate both beakers at about 40 degrees then, and leave it for about 15 minutes. The next step is to filter the contents of both beakers and then record the volume of juice that you get in each. You should find that you get much more juice from the beaker containing pectinase than the one with water.